Okay, we're moving on to publishing and sharing.
This is where you get to retaliate and ask me questions. So the issue that we're focused on, you've heard a lot about today in the Science Exchange talk. The reproducibility project cancer biology came up, and that's actually what I start with. And one of the people that I quote is Manya Baker, who is here and wrote beautifully about it when the first result in eLife came out in January. And Ed Young in The Atlantic, talking about the difficulty of reproducing previously published cancer papers that we already heard about, what he wrote was, the hardest part by far was figuring out exactly what the original labs actually did. Scientific papers come with method sections that theoretically ought to provide recipes for doing the same experiments. But often those recipes are incomplete, missing out important steps, details, or ingredients. In some cases, the recipes aren't described at all. Researchers simply cite an earlier study that was used in a similar technique. So Science Exchange had to chase down what exactly was done, what's the protocol, and as Manya wrote, in many of those cases, the people who did the work aren't there anymore, and somebody else is looking through their lab notebooks. We can do a lot better than that. And so one of the things that we're trying to address is move us away from contact author for details in the materials and method sections and the ubiquitous, we used a slightly modified version of the technique reported in another paper. Good luck figuring out what was modified. And part two of what we're trying to do with Protocols.io is centered on the absurdity of my own postdoc experience, where I spent literally a year and a half on this protocol right here, and whoops. It took me a year and a half to figure out that this step here, instead of a microliter, needs 5 of a particular chemical, and instead of a 15-minute incubation, you need an hour, which is a wonderful year and a half. But the crazy part isn't that it took me a year and a half. The crazy part is this is not a new method. This is a correction of something that's previously published in Nature Methods, which means that there is no publication, right? So A, I get no credit for this year and a half, and B, and that's the more devastating part of this, everybody else who's using this technique is either getting completely misleading results or has to spend a year or two to rediscover what I know, what I would love to share with them, but there's no infrastructure. There's no way for me to get that modification out, and I can't go to the Nature Methods and put a little post-it note on that part of the protocol. So in 2012, we got started with creating Protocols.io, which is an open access platform that allows you, A, to get these details out in the first place when you're publishing, and B, to keep them alive, version them, and to have them evolve, because science really does not stop with a publication. I'm not going to do justice to the gorgeous platform, both on the web and the native iOS and Android apps that we developed over this time, because I don't have a computer here to actually demo it, but you should go and take a look. We put a lot of thought and effort into the user experience, so it is a beautiful platform, but just a few examples of what we have now. So there's about 6,000 protocols, 2,000 of them publicly shared, 4,000 private. You can use it either way. A lot of them start out privately, and then as you publish, they move into the public space. And so a few examples just highlighting this. Here's a protocol from a student in Australia, and someone using the protocol. This is a DNA extraction technique from plants. Someone using the protocol noticed that in this step 24, there's instruction to dry the DNA for somewhere between 2 and 2 minutes, and there's a 7-minute timer. So somewhat confusing instruction. The person asked the question, what do you mean? The question instantly goes to the author of the protocol and anybody else who's using it. And then the author responds, creates a new version. So there's now version 3. It's a 2-minute timer. It should have been 2 to 3 minutes, not 2 to 2 minutes, and responds to the question. So on step level, you can actually ask a question. It'll go to everybody who's using it, and you can have conversations. And this is that post-publication part, and you can create new versions. Or if you're not the author of the technique, you can create a copy of it for yourself and then optimize it and share what you've done, and people can compare side by side the different methods. We are partnering with more and more publishers to, as I said, move us away from contact author for details. So here's a PLOS One paper. We partnered with PLOS back in April. Here's a PLOS One paper that has the code on Code Ocean. You'll hear about them in just a minute. Runnable code has the protocol on Protocols.io with videos inside. So it's not just text. You can put images, videos, right? And this is really where we're moving in terms of publishing. It's not just a PDF. It's not just a story, but it's an ensemble, right? It's a puzzle that you're putting together with different objects that you need to reproduce and follow up on the work. And finally, my last and absolutely favorite example in this question from a researcher in Chile: how do you get RNA out of neuron cultures? Postdoc from UCSF starts responding that you should use RNA trisol technique. That should work. I know that we have 5 or 6 of them on protocols.io. And I mentioned that, and the UCSF postdoc, she took a look and said, oh, this one should work. And what I love about it is, this is actually, I took a look at this protocol. This is a protocol from a paper, not in PLOS, but in the journal GigaScience. That paper focused on parasites of stickleback fish. Has nothing to do with neuron cultures, right? So because that protocol is in a public repository and is not contact author for details, but is out there, someone who needs it for neurons can now find it. So it's not even just the reproducibility in the paper itself, but the impact goes far beyond your own publication and your own use case. And this is the number of journals that are starting to include us in the instructions for authors. Genetics, GigaScience, and Public Library of Science were the first. And since PLOS announced it back in April, there was 10 journals in April. By July, there were 50 that had us in author instructions, about 200 today. And so we're almost done, just like 26,000 more to go in PubMed. And this is the number of scientists on a monthly basis who are starting to create protocols. So it's starting to change. There's a lot to do, but there is good progress. It's open access, free to read, free to publish, and people who want to learn more about the business model can see me after this or ask a question about it. And I know I was supposed to stop. This is where we are today, but where we're going in the future is connecting the app to the actual devices, so that if you're about to spin the centrifuge at the wrong speed, right, the app and the centrifuge can talk to each other and can tell you, this should be 3,000, not 12,000, you'll burst your cells. So that's where we want to go further on. And there's a remarkable team that I didn't do justice to because I didn't actually show what they've built. Questions? Manya? There's a microphone.
Oh. Which communities are most open to this and most excited about this, and which communities are you having trouble getting traction with?
So there is a bias. We've started with lab methods, and then we expanded it to computational. There is a natural bias towards genetics and genomics just because I came from that community, and those were the earliest adopters. It's not so much that there's resistance. I think we need to do a lot more for chemistry in terms of what the protocols supports. So there's a lot more work that we need to do. We don't have chemistry protocols, but particularly with the partnership with PLOS One, because that's so broad, that has started to expand. We're starting to see psychology protocols, medical device protocols. I haven't seen really resistance. It's more a matter of awareness and who knows about it. So there is a bias towards the genetics genomics, but I'm not sure that it's because of resistance from other fields.
So Lenny, I'm going to be really annoying here. This is Anita. When are you putting in RIDs, as the funding overlords have asked?
We are trying. So this is the RIDs. You'll hear, are you speaking? Yes, so you'll hear more about them. This is part of the effort to make it clear what is the actual reagent that was used. We've started adding vendor catalogs. We're talking with you. There's just a backlog in us processing, getting those catalogs in, and you have a lot of reagents that we need to get in, so that's part of the backlog. But let's talk more offline. That is clearly a great thing to do. I'm out of time, so Mike, we should talk offline so that we can.
Just with Anita's question there, is there going to be required fields to make it more useful for other researchers, like lot information or RRIDs?
Oh, that is I would love to spend another 10 minutes talking about what you require in a protocol. We don't have requirements other than the author name and your affiliation, pretty much, right? And you need to give it a title. We're not in the position. We need adoption. We need scientists to like us, and we need to be flexible enough. If we start telling you it's not going to take you 15 minutes, but it's going to take you 40 minutes to put in your protocol, and you have to detail the version of the software that you used and the catalog number of the reagent, that would be wonderful for reproducibility on the one hand. On the other hand, we'd get nowhere. We'd have no adoption, no journal partners, and no scientists actually sharing the protocols. It's a very tricky balancing act that you want. So you want to encourage people to do it, but you can't force them. That's why partnering with RRID, getting the vendor catalog so that you don't need to spend much time putting in the reagent, but you can just click on the catalog number and it's in that step. Making it easier is the way to go. I can't require it from the scientists.
All right, guys, thanks for having me. My name is Alberto, and I'm with Authorea. So for the last couple of years, I've been repeating a mantra over and over to try and illustrate one of the problems with openness and reproducibility in science. And it goes like this. Scientists produce 21st century research, but they write it up using 20th century tools, and they package it and publish it in a 17th century format. So I'm going to walk you through that step by step, and I'm going to try and do it in just 1 minute. So I think we all agree on the fact that scientists produce new, cool stuff. It's cutting edge. If it's not new, then it's not scientific research in the first place. So by definition, science and research is 21st century. It's also becoming more computational, data-driven, collaborative, automated, web-based, and I'm very happy that all those trends are happening in science. But if you look at the tools that every researcher or most researchers are using to write up scientific research, they're using a tool that you can see in the top right in its full glory. Nobody has talked about this tool. It's the most used scientific app in the world, and it's called Microsoft Word. You see it in its full glory with track changes enabled, which is a nightmare for a lot of people. So what's the problem with these tools? From PDF to LaTeX, that probably some of you know about. I hope not. To Microsoft Word, Excel, PDF, all these tools were built and designed in the 1980s when most researchers were working offline and alone. Now we're working online and together. So they're not data-driven, they're not collaborative, they're not version controlled, and also they're incompatible. I mean, if you ever tried to convert a PDF document to Word, you know what I'm talking about. The most depressing, tragic, I would say, part of the story is that we're still packaging research in a format that looks like that. That paper is a few hundred years old, doesn't look too different from what you see today in PNAS or JAMA. We're publishing the same stuff. PDF is just a photograph of a piece of paper. Some computer scientists may disagree with me, but it is just a photograph of a piece of paper. So we've advanced science for 100 years, but we're still publishing pretty much photographs of paper. So yeah, they're static and lifeless. So what's the problem? I think that my main point today that I'd like to bring across is that scientists need to publish, because that's how they get credit, recognition, tenure, community recognition. So until we have an academic publishing system that is built around publishing PDFs, we can talk as much as we want about data and openness and reproducibility, but at the end of the day, the scientist only cares about the final publication. And the final publication, if it's static and lifeless and doesn't contain any data, then it's hard to make work reproducible. So I'd like to propose that the real barrier to open, reproducible research is not Elsevier. I'm actually also interested, and not many people have mentioned publishers today, but yeah, I think it's Microsoft Word and PDF. That's the one thing that is creating a barrier towards openness and data-driven, reproducible research. So we've been building a tool. It's called Authorea, and we're pretty much building and rethinking the document, the scholarly article of the future that I think should be data-driven, open, version-controlled. We're built on Git so that every single document you start is a new Git repository. It's also web-based because everything we produce today needs to be HTML first. Format neutral, you can write across different formats. We're really rethinking a scholarly document to be an API, something that you can connect to from different sources. So this is the point where I normally have a demo. So if you can start a video, like Lenny said, I would have loved to give a demo, but it wasn't possible. So instead, I'm going to show you a video. If you can also full screen it, that would be great. So this is pretty much how the product works in 3 minutes, and then I'll be done. So as you will see that it's a document editor. So you'll see that I just created this a couple minutes ago, and it goes super fast. You create some content. You have a toolbar at the top. Then you can use the toolbar options to just highlight stuff and make it bold. Then I'm citing myself here. I'm just searching for my name. Found a paper that I wrote with Michael Kurtz. I cite it, and as you can see, the citation and the reference gets created automatically. I can also cite the work of a colleague of mine, Josh Nicholson. I could search by keyword and all of that. And then very quickly, I can also change the format. And once again, the bibliography is created on the fly. This actually saves a lot of time. Now, important part, I add a figure, and I have a figure that is like from an old astrophysics paper. I can, it gets numbered. I can write the caption. I can change the size. And then importantly, I can add the data that is related to the figure. And on top of that, I could add a code. So if I have an IPython notebook, a Jupyter notebook that goes with a figure, I can actually add it. And I'm not going to show it today, but you can execute the entire Jupyter notebook. You can also add some LaTeX for the mathematics people in the room. It looks nasty like that, but when I click outside, it looks beautiful. I can also do things like referencing different parts of the document, because the document is dynamic and semantically structured, so I can refer to a specific equation or a specific figure, so that if you add another figure before figure 1, you don't have to rename all the figures. You can add things like code snippets. There's a new thing that we just added. You can add a code block, and we automatically recognize the syntax, whatever language you used, so it renders beautifully like that. So as I said, syntax highlighting is automatic. And then a couple more things, and then I'm done. You can highlight some text and add a comment to it. Kind of looks and feels like Medium in a way. So you are able to reference different parts of the document by using comments. You can resolve comments and use them to talk to your friends or enemies. That's the table of contents that, of course, is created because the entire document has semantics, so it's structured in a certain way. And this is the killer feature. We have word count as well, which is dynamic. It allows you to count words. If I delete a sentence, it's in the history, because everything is version controlled, it's Git-based. So if I go back to a previous version, you can actually see the part that I removed is in red, and I can even revert it. And finally, if I want to send it to a journal or a preprint server, such as the bioRxiv, which we're integrated with. We're integrated, I think, with 10 publishers at the moment, 25 publications. You can send it in just one click. Or if you need a good old PDF, you can do that as well. And the very generic PDF looks that way. Or you can just create a different type of PDF by changing the parameters. So you can actually change the look of the PDF on the fly. So here, even changing the citation format to be a different style, from a journal of brain behavior and evolution, and there you go, you download the document, and now it looks different. Final part, we're integrated with Jupyter Notebooks, but also with any arbitrary JavaScript format like Plotly, D3.js, so that's an example of a Plotly graph. And I think I'm really out of time, so I think I'll take one question. Thank you so much.
If nobody else has a question. Are you actually writing this in JATS 2.0, or which JATS are you in?
Yeah,
and
so for the time being,
convert?
Yeah, so for the time being, when we ship the content to publishers, we give them PDF and XML JATS, which is the publisher standard for receiving content. Going forward, bioRxiv, eLife are two of the very first publishers we're working with to send them no PDF at all. So we'll just send them native XML content so that they can, as you know, probably bypass a lot of the typesetting work that they do, which is slows down, I think, research publishing a lot. Cool. Thank you. Oh, Lenny, okay. Can I take it? Yeah? Okay.
It's not a question, it's just a 10-second comment.
Yeah.
I've been writing a paper with 14 co-authors using Authorea over the last few months, and it's just wonderful.
Thank you. Thank you.
Hey. Hi, everyone. Can you hear me? I'm Code Ocean. I'm the CEO of the company. And I want to start with this sentence over here, which I heard from a fellow of mine from the Computer Vision Society. We were sitting for lunch, and he was saying, you know, Simon, it's very simple. If it's not reproducible, it's not really science. And I think that's the gist of it. And I'm going to go back to that after I'm going to talk about Code Ocean and how we started. So I did my PhD at Tel Aviv University and worked with the German Space Agency. And we were going to the field, collecting samples, ground truth samples, having satellite imagery at the same time, and we were starting the area for environmental impacts in coal mining areas. And I want to tell you a secret, we were not the first ones who actually studied that specific field. And I was looking to the literature and going through hundreds of articles to find the state of the art to generate something like this. Sorry. Okay. Something like this. And when you look at articles, you basically, you see different types of results. What you see here are different algorithms finding the changes between 1 year to the other, and generating those images. Each and every heat map here is a different algorithm. And for me to go through the process of generating all those images, let's see if it works. Yes, I need to go through all those steps. So find the code. If you read the article, in many cases you will not see any mention of code, but if you look for it on the web, you will find it. And so finding the code and then acquiring the right hardware. In some cases, you need specific hardware like GPU or very large amounts of memory. So you need to find the right hardware, and then setting up the environment. You have different operating systems. I'm using a Mac. Somebody else uses Windows, Linux. You have different programming languages, Python, R, MATLAB, and so on. And then after you set up the whole environment, you need to have the right files. I remember taking a research project code that was hundreds of files. And only 10 files were actually the ones that you had to use this algorithm. So find the right code files, and installing, debugging, it takes forever. Sometimes it's not really successful. It's a real pain. So we at Code Ocean, we want to eliminate all those steps here and just have this run magical button so that every person, researcher, who wants to use the code that you read the article will be able to just run it, not install it. So Code Ocean is an online code execution platform that integrates with any scholarly platform. It can be a publisher, it can be a preprint, it can be any other notebook or so. And what you see here is a template of of a publisher website. You have the name, the authors, abstract, and then we build this technology, which is a widget that you can add, and at the same place that you read the article, you'll be able to run the code. You can access the code, download the code. It's all open access. On the CodeOcean platform, you can discover, execute, and collaborate as well, just like Google Docs. This is the current collaboration that we have. We're integrated with the IEEE, which is the biggest engineering publisher in the world. GigaScience, F1000 Research. Tomorrow we are going to announce a new collaboration with the SPIE. It's another engineering publisher. And many more to come. We're in the pilots with 75% of publishers. Going back to computational reproducibility, fundamentals of the research scientific process is skepticism. And the reason that we have peer review is that we need to verify that a claim can be independently verified. And the research community is a communal place where we build on top of each other's works. But in order to do that, we need to share the work so that we can build on top of it. If we don't share the work, we will stay with skepticism and we will not be able to build on each other's works. There are different levels of reproducibility. No reproducibility, where you have only publication. Full reproducibility, when you have linked executability, code and data. This is the golden standard. This is by research published in Science. This is some data that was published by Victoria Stoughton in PLOS, where she looks at different adoption policies. We're talking about biopolicy, but in general, policies around data and code. In 2011, 2012, the first two rows are talking about requiring the code and data, and then encouraging, implied, so it's good to have, or there's no mention. And you can see the growth in the last few years, from 2011 to 2017 with data and code. Code is still lagging behind data, but it's reaching a point where there's a certain amount of journals who actually require it, but the big growth, I think, is around encouraging, which is a big change in the last few years. Really. This is the ecosystem here. In the middle we have the researchers, we have funders, scientific societies, university, regulatory bodies. We all need to come together. It's a big thing for us to reach reproducibility, and there's a lot that we can do. I hope that in the panels we can talk about it a little bit more. Touching upon the example that Lenny was talking about, in the scholarly record, he showed an example where a research article, protocols, code and data in an executable way are coming together. This is, it's not going to play if we don't have all the missing pieces in this record. Think about a music record, it's not going to play if you're not going to have all of this and the infrastructure to actually run it. This is my last slide, almost the last slide. This is an example of a change that the Psychological Science Journal did, introducing open data badges. And you can see what happens here from an adoption of less than 5%, reaching almost 40%. So this is an example of things that we can do as part of the ecosystem to reach more open data, open science, and reproducibility. And this is sort of the last quote I really like. This is talking about biology. There were a lot of talk about automation and robotics. we need to remember that the software that people are doing in simulations, contains ideas that enable biology. And we just want to make this thing accessible for everyone as well. Thank you.
Hello, thanks very much for inviting me.
My name is Meredith Morovati. I am the Executive Director of Dryad, and I'm going to talk about a few examples around open data publishing. Let me get my things in order here. This one. Oh. Okay, got it. Thank you. Okay, so the idea of open data usually infers publishing data that supports an article or some kind of scholarship, and this is something that Dryad, the organization I represent, focuses on. The idea here is behind the article is the main argument and scholarship, but that the data itself are the bedrock of this, and that all data that supports an argument in a paper should be made open for reproducibility, for transparency, and so on. Dryad is a digital repository that links data to scholarly articles. This is a screenshot of the home page and the location where you can submit data in the upper right-hand corner. Is there a pointer? We have over 18,000 packages in the Dryad system, and a data package, by our definition, is a data set that links to one article. These are usually made up of about 3 or 4 files on average, but can have many more files than that. And we have data in our repository that links to over 640 different journals, and this data is being used and viewed all the time. We have over 2 million downloads of data in Dryad just recently. So Dryad offers a technical integration with journals or publishers or societies to link data to articles in a more automated and supported way. We have accomplished this with over 100 journals and with many manuscript systems. Integrating is when we set up the sharing of metadata between the manuscript submission system and the Dryad data submission system. It's a technical workflow enhancement that takes advantage of the timing of publication to also capture the data. We know that if you ask an author for data after publication, it's often very difficult to receive it. So during the time of publication is the right time to ask when their attention is on this task. So the automatic setup has many benefits. It provides ease of use to the author. It frees a publisher or society from having to collect the data, manage it themselves, various data files and types. Now they can just point to a digital object identifier. And we also offer workflow enhancements with our integration. Here's an example of what we call the review workflow. By integrating data submission system with the manuscript system, we can offer some automated sharing of article and data set metadata, and we can provide an opportunity to support the data privately during the review process. Reviewing data is an important consideration to take on. About 50% of the journals that are integrating their manuscript submission system with our system are choosing to use this review data workflow. Now, review processes for each journal are vastly different, but the general idea is to at least check some basics and review metadata or methods or whatever the key items in the data are that that journal or society is struggling with. Dryad publishes open data with all fields. And though we have an organic beginning in the life science and genetics and evolution, we do now manage data and support data from all fields. So therefore, our setup and our requirements need to be very flexible and customizable. It needs to serve all kinds of communities, and they all may have different needs or challenges with regard to reviewing data. But you can still perform some basic checks to make sure that the data that you have meets your expectations. Publishing data openly is essential for the reproducibility and transparency of science, publishing data openly. This is an example of a BMJ open study of prostate cancer associated with the use of oral contraceptives. Researchers were looking for a link between these conditions. This was published in August of 2011. But it was found, when others were able to access the data, that there were some accidental miscoding, and the paper was corrected in January of 2012. So you can see some of those screenshots here. It's possible that a careful reviewer might have caught this. BMJ Open was actually integrated just before this article was submitted, and they actually do use the review workflow, but they probably didn't look closely at the data at that time. Reviewing data is not that common now, and 6 years earlier, it wouldn't have been common even for a progressive publisher like BMJ Open. Very few journals, some do, but very few rerun the analysis in a way that would identify these common problems at the publication stage. However, we do know that reviewing for metadata, making sure data fits the expectations of your journal, catch errors. And importantly, the open data is what enabled the self-correcting nature of science in this case. It was publishing the data openly. So this outcome is actually pretty positive. A mistake was found, it was corrected, and it was updated. However, there are more examples of how a study was found to be manipulated or fraudulent when other researchers try to recreate a portion of a study or review it critically. On this slide is an example of a couple of different examples. There's a massive social science study where a survey portion was completely fabricated. There's an example of falsifying compounds, an experiment and using incorrect measurements in a nanoparticle synthesis. The nanoparticle paper example shows that when data are not provided, and it takes a long time to receive this, it can take many years and many thousands of dollars of resources to correct the information out there. So essentially, open data is essential for reproducibility and for transparency. But so can reviewing data at various stages of article acceptance. Increasing the amount of data you review will help researchers catch small mistakes, could catch some things earlier. Ultimately, science will not be reproducible if the data is not available. And open data supports all of these things. Thank you. Did anyone have questions? Everyone knows open data.
Number 1 or number 2?
1.
Okay. All right, that looks reproducible. Earlier today, I actually had problems getting that title up. So, all right, so today I'm going to be talking about key biological resources and the RRID system. My name is Anita Bandrowski. I run SciCrunch. I'm the founder and current CEO. Now, no one seems to be able to figure out the clicker, and that includes... What was that? Like that? Ooh, look at that. How cool is that? Okay. All right, so now this project was actually, it is about 10 years old. It ran out of a couple of really big pan-NIH initiatives. And the current stage that we're in is the SciCrunch Inc. portion, which is essentially making the thing more robust and commercial grade. But a lot of work went into it in the academic side. We've heard a lot about this. I won't belabor this point, but protocols and our scientific papers are really like a recipe, right? So with a recipe and with any scientific recipe, we'd like to ask two really basic questions. What things were used? And what other studies have actually used these same things, right? So Why would we want to ask this? Well, reproducibility we've heard a lot about, but what we haven't heard a lot about is tracking. Tracking problems, especially monitoring the health of the scientific food chain. I mean, if you look up there, we can figure out, which farm was contaminated. We can't do that for antibodies. So why can't we do that now? Well, here's an example. Now, you can text mine the heck out of this if you want to, and that's great, but you're never going to get anything better than we don't know. Right? We don't know. This vendor does not have that antibody. We can guess what they may have meant, but we don't know. That's a problem because we don't know a whole lot. So we don't know more than 50% of the antibodies. That's pathetic. Okay, that is truly pathetic. Cell lines, constructs, organism, all of these things have problems. And this is the simplest part of the study. Which mouse did you use? Do we have an answer for that? Usually yes, but sometimes not so much. All right, so can we fix that? Turns out that this is actually a fixable problem. It is an amazingly fixable problem. We've been working on this fixing for a while. We just did a lot of talking for about 5, 6, maybe 7 years. We brought on a whole bunch of people, and they were, gray and whatever, and they were very important. And they had all kinds of stakeholders and everything else, and nothing happened. Nothing happened. Until actually it was 2012 when we finally got a meeting together with apparently the right people, and we actually started a project called the RRID Initiative. And what happened is we actually got a pilot with 25 journals, because 25 journal editors agreed to come on and do this pilot for 3 months. All they gave us was 3 months, and then no one stopped. That's how you know you have a successful pilot project, because no one actually stopped doing this. So, what are RRIDs? It is the central barcode. It's the scientific barcode for reagents. Some companies actually reflect the RRID. That's a mouse, and that's an antibody. The scientist will buy these things, and then when the publishers go in, and when the scientist actually publishes, then the publishers will ask for these RRIDs, and then what that allows is a whole lot of linking of data and other things, as long as you know how to use it. So here is Science Direct, which has a little antibody app, and they actually link through, as does eLife, to some of our services. Now, what that allows is a whole lot of interesting things. But let me just show you what this RRID looks like, and why do we need yet another way to identify these things, right? So we have names. Aren't those good enough? Who can tell what those things are in the room? Just show of hands real quick. Yeah, no one. Okay, that middle thing is a mouse, because it's a black 6. And the top one is actually a fly. Cool, huh? But in fact, the RRID is a globally unique catalog number. It's really simple. But they answer really different kinds of questions. You know, the regular catalog number, what can I buy and what can I buy today? Whereas for the RRID is actually unique across time, not just current iteration of a catalog. So that's the anatomy of an RRID. It's this resolvable entity. It's resolved across a whole slew of resolvers like identifiers.org, N2T resolver, and of course us. And it tells you what this is. In fact, when you look at the whole syntax, you get this company or provider, you get the local identifier, you get the version or lot, which we suggest. We get very few of those. And then, of course, the global identifier. So what should authors actually do with it? The workflow is so simple, I didn't need a video. The journal says, go to this website. They put in their particular search term up there. It's actually relatively simple. They put in 108L, that's what I put. And this is for an ambystoma, which Ambystoma mexicanum is actually a critter that people use. Then what they're able to do is click on the Cite This button, copy that thing, and just paste it into their manuscript. And magically enough, the paper is published and it becomes data. How cool is that? Authors seem to like it. This is from a few authors in eLife from a blog that we published. They said, hey, this was actually pretty easy to do. And the first author up there, talked about it will actually help members of my own lab track down what we did, which is kind of nice. And then it becomes really important, because we have things like problematic cell lines, and these are notes and comments about the cell line which is contaminated at the vendor. These are contaminated at the vendor. Do you check this every day? There are hundreds and hundreds of these things. No, you don't. Many labs just go ahead and buy this and use it, and unlike ATCC, some commercial banks do not tell you that these things are contaminated. So you can buy this, do your study, you can still reproduce it, but it's not the right cell line. That's a problem. So what we do is we do a bunch of aggregation of this data, we put it all together, we actually have a master copy so that these things are actually completely persistent, which is very different. Some things are no longer in service, so we just make a note of that. And the end of the story is that, does this actually work? Well, yes. At least in that pilot phase, 90% were identifiable when you looked at antibodies or organisms or tools versus 50%. And this is looking at the same journals across the study. And what's actually on there also is that 97% accuracy was achieved by the author. By the author, 97% accuracy, which is amazing. Some journals, like eLife here, actually link through to us to the resolver, and what we're trying to prevent is that first case. ERC2 mouse from Jackson Laboratory in Bar Harbor, useless piece of information, but there's the stock number. That's much better. That's what we're trying to get to. Okay, so resource creators, I'll go really fast. What we're doing is democratizing this process, which allows us to actually not have to use fancy tools, although we do have lots of fancy tools. And there's one of them, it's actually 15 lines of Python, which is our little robot reader of these articles, and it brings back data about the resources used. Thank you. Questions? Yes, sir.
Hey, I think this is awesome and amazing, because one of the problem with publications is scientists don't even, they just write the protein and the vendor. They don't even write a catalog number.
Exactly.
But having that a reality, how do you get them to do this? And Len was actually the conference we had in Toronto, and one of the things scientists said is, why aren't you guys forcing me to cite stuff? So how do you get us to all the publishers and force them to cite, scientists to cite?
So currently we're at the point where we have about 350 journals where we have actually found RRIDs. We have no idea how many are enforcing it effectively, but Cell Press is enforcing effectively, as is eLife, as are a lot of the society journals. So we keep working with people to get more stuff, and then we get I don't know why some of the Nature journals actually, like Nature Communications has a whole bunch of papers with RIDs, and we can't figure out why. But somebody must care, because this is something that can be done without any additional publisher workflow. So it's just, if somebody cares, they will put them in. And we don't know why. So the thing is growing. It's not up there anymore, but you know, the number of papers that we get every year is kind of hockey stick, which really begs the question of scale. Because now we're at the point where it's really important to understand how we're going to scale.
It's awesome.
Thank you.